Yeah.
Like, so as we get older, we start to lose our ability. Well, we lose sensitivity to a lot of things, like vision, hearing. We start filling in the world with memories. And so we don't actually see the world for what it is. So a little child, like 5 years old, you put identical monkeys in front of this kid, he'll be able to point out the differences. And you put an adult in front of these monkeys, and they look identical. And the same thing with hearing. We become desensitized to the world. We start using our memories to fill in everything. And so we start not being able to hear the higher frequencies. And that's why I've heard the students will set the ringtone to these frequencies that only they can hear. And so the teacher, the students will hear it, but the teacher can't. And that way the student will know if they're getting a phone call or something. But yeah, low sounds like growls, our survival sensory organs allow us to hear those. And the high-pitched stuff, we didn't really evolve to necessarily need to hear. Some of it's really a survival thing. None of this will be on your exam, by the way. But that's the things we talk about. And we actually have a YouTube video where some of the students actually can hear all of them. So if we maintain our sensory organs, then we can keep that more. We can still, like I wrote, like, are you still a child at heart? Because, you can kind of maintain some of those capabilities.
Does doing like vestibular exercises like balance sports help maintain?
I don't know, and I don't know if anyone's ever looked at that. But I would imagine just being present instead of getting caught up in your head all the time. And as we get older and older, we just start to rely on memories instead of being present. And I've had to work on that since graduate school. I got so stuck in my head. Don't do it, guys. My intellect became like a prison for me. And so I've spent the last, 10 or so years of my life trying to maintain awareness of just the present moment and not in my head, like doing this and that, and it will serve you even for biochemistry. If you can stay in the classroom in the present moment, then you can actually learn the material better. It's like I was anxious. That actually kept me from learning in graduate school. And that's like students come to me and they're nervous and they're worried. That's actually keeping you from learning. You gotta have to have like a relaxed, kind of more passive state as you're in this class right now, because you're gonna absorb the material better if you can kind of relax your physiology. Being resistant is not gonna help you. So I'm gonna try to relax. And just whatever questions you might have, like as students know, if you haven't been in here before, it's like you have to put in a couple of quarters by asking a question, and I will just go run around on the board to try to help you understand whatever it is. So do we have any starting questions?
Yeah, can we get like an overview of like study that does this like specifics, like highlights?
Like an overview of what so that's a broad one. Like I need to be narrowed down a little bit. Are you talking about after the last exam too?
Yeah, kind of. So like the material we've covered since the last exam, like the highlights of those ones, like specific stuff.
And so I like to I also use my computer so I can kind of remember what. So since the exam, so I'm going to start with since the exam, and let's focus on that material. So starting with week 8, so we started with nucleotide metabolism. So nucleotide metabolism, and this is where we're focused on the synthesis and breakdown of purines and pyrimidines. Wait. Was there another one before that, though? No, it wasn't this one. So the synthesis and breakdown of purines and pyrimidines is kind of where we started. And I'll just write them both here, and then we'll go from there, see what you guys want to focus on. And then we moved into DNA replication. stuff you guys need to know with the DNA helicase and DNA polymerase and topoisomerases and all of that really fun stuff if you guys remember. So that was the second part that we went over. And third part would be RNA transcription. That's what we started with last week. And we then went into RNA translation. And that's where we're going to be making a protein. So we're going to make a polypeptide with that one. Whereas transcription is where we take DNA and we make messenger RNA. And then we left off yesterday with what? Regulation. So gene expression regulation. That is like the really big overview of the new stuff that will be on the final exam. And it'll probably be 60-40. That's usually the way I have it set up. New stuff is 60%, old stuff is 40%. So what of this feels the most, I don't want to say intimidating, but feels like it needs the most review? Is that like, Savannah, just a general breakdown?
Yeah, so like, for nucleotide metabolism, like, steps.
Yeah, and as I said, like, don't kind of start with the products or with the things that are going in and the major products. All that stuff in the middle, I told you guys, like, don't memorize those enzymes, don't memorize all those intermediate steps in the middle, but you want to know the starting material for purines and pyrimidines and the final material, what we end up with, which are the nucleotides. That's what these guys are. We're building nucleotides, so we got to make the nitrogenous bases first.
Yeah, and so for DNA replication, like the enzymes.
Yeah, so we start out with helicase, and then we have topoisomerase, the DNA polymerase. And these, to me, when you're studying these, like just thinking about, if you just know the function, just have a list of these terms, and there's even a table in the slides, and their function, then you can start putting that together. And hopefully with the lecture, like maybe you guys, like what's the thing that stops your DNA from doing that? Slapping back together.
Yeah.
Single stranded binding proteins. And that's really, DNA replication is really kind of that.
But is it 3 to 5 or a 5 to 3, Doc? That's the question.
Exactly! What is it, Freezy? Tell us. Ignore him! It's the opposite. It's 5 to 3. I'm guessing he knows.
So just like steps for RNA transcription, DNA, and RNA translation, and then gene expression, like factors that affect it, and then this replication stuff.
Yeah, I mean, yes. If we want, we could do like a whirlwind of just, we've done this before in this tutoring where we just kind of go through all of them and we see how much we can get through for each one of these, like starting with the first one, nucleotide metabolism. And I usually just, I like to take any specific questions, but if anyone doesn't have specific questions, then we can just go through starting with the, again, when we do this, like you still have to study your notes, but I give you like the big picture stuff to focus on. And we could start with that, with the purines and pyrimidines. You're saying yes or no?
No, that's
Yes.
the question I would prefer to go over stuff from yesterday because that all just kind of slid off my very smooth brain.
Do you want to start with gene regulation? I guess it doesn't matter which one we start with.
Pyrimidines are mostly just pure memorization, but it's not that bad.
I actually just made the review slide for that, so for the gene regulation, I know this one was kind of like, it maybe might have felt a little more loosey-goosey because there's just a number of different things that we picked on depending on that. So with gene expression, there's lots of different types of gene expression depending on the level. So you can have starting with epigenetics. This is kind of one of the first ones that we talked about. So this controls whether the genes can be accessed. Whether or not genes can be accessed. So what does epi mean? Above. So epi means above the genes. And so this is the methylation or acetylation thing. So with acetylation, you have, they're called histone acetylation transferases, or HDACs, which are histone deacetylation transferases. So they put on or take off acetyl groups, versus a methylation is just a methyl group. But methyl groups will silence a gene, so you can't even access it, versus acetylation will give access to the gene, and HDACs, when you take off the acetyl group, it'll prevent access to the gene. So that's kind of the first level of DNA regulation, or gene expression regulation, which is what this is about, regulating gene expression. So it has nothing, it's not really about regulating the gene itself, but whether or not the gene can be accessed. And then we have also and post-transcription regulation. So this would, the examples we gave for so one of them, pre-transcription regulation, the example was for prokaryotic cells. So this isn't part of eukaryotics, but it's a really important part of prokaryotic cells, have operons. And I can't write it all here, it would take way too much time, and I have a lot to go over. But what are operons? I have the definition in my slides, but they determine that there are a group of genes that are going to be regulated together. And the most common one that we talk about in biology is the lac operon. So an example is the lac operon. And these are all genes that code for lactose enzymes. And they're transcribed together. And so they're going to be regulated together. And there are repressors that can bind to these operons. And the operons are just part of the promoter regions. But this is a form of pre-transcription regulation in prokaryotic cells. So if a repressor binds to the operon, it's going to turn it off. If an activator binds to the operon, it's going to turn it on. And all those genes for lactose will be transcribed at the same time. And that's how prokaryotic cells regulate gene expression. That's really it. They just have operons. Eukaryotic cells have a lot. So we have epigenetics. Eukaryotic cells have epigenetics. And then what we talked about for actually we didn't talk about, we talked about enhancer regions. So for eukaryotic cells, we talked about enhancer regions and transcription factors. These will regulate whether or not a gene's expressed. So if your TF2s, remember that what are TF2s? These recruit RNA polymerase. That's a type of pre-transcriptional regulation. If those transcription factors aren't there, if the enhancer region's not enhancing transcription, all of that is gonna delay transcription. That's a way to regulate eukaryotic transcription. And that's a pre-regulation, a pre-transcription regulation. The post-transcription regulation we talked about are spliceosomes. Splicing of exons, introns. And that's a type of post-transcription regulation, because remember, splicing happens after the RNA transcript is made. And this is another way you could regulate whether or not a gene is expressed. When you splice that mRNA, you're affecting that gene and how it's expressed. And I think that was, and the other one we talked about that's post for eukaryotic cells is 5' cap. That's a 5'7-methyl-guanosine cap, and the poly A tail. And these guys are provide stability for the mRNA. So in other words, if they aren't there, that mRNA is going to be degraded more easily. Which means that's a way to regulate expression. If they doesn't have a 5' cap or a poly A tail, then it's going to be degraded. It's a regulation. And that's a post-transcription regulation. So there's pre, this is regulating whether or not the gene's even transcribed, and then post, or the gene has been transcribed, but now what are we going to do with that mRNA once it's been made? So that's after it. And so that's why there's a lot of different types. There's more than this, but those are the examples I gave. Then we also had pre and post translation. And the examples I gave those were for pre-translation, I gave the example of eukaryotic initiation factor 2. And this guy, it binds to the ribosome when it's active, and it's active when it's dephosphorylated. But when it's active, you don't have to know the phosphorylation part.
What did you say the EIF was?
Eukaryotic initiation factor 2. When active, it binds ribosome and helps match mRNA codon to the tRNA anticodon.
Can I tell them how I remember?
Sure.
That was interesting. Think about it, it's like a matchmaker, and you're trying to put together the mRNA and tRNA so they can fight each other. And if you think about eHarmony, if it works, tRNA and mRNA fight each other. Easy.
So whatever works for students, that's the gist of it though. And the other one that we talked about was post-translation, and this one was ubiquitination of proteins. And there you go. That is regulation in a nutshell.
Is IF for interest? It's not.
I stands for initiation.
Initiation. It's another I or another.
Eukaryotic initiation factor 2. And it's just one of those protein. We learned that there's a lot of protein factors. I did make you guys memorize all those. That's the only one that I use as an example because it helps regulate. All this regulates gene expression. So when you ubiquitinate a protein, are you tagging it to survive longer, or are you tagging it so that its life is cut short? It's destruction. So it's when you ubiquitinate a protein, then you get rid of it faster. That's another form of regulation. If a protein sticks around longer, it's going to keep doing whatever that job is and that's gonna change the cell's function. If it's, depending on for example, hepatocytes, your liver cells, they need a lot more enzymes for making cholesterol because that's their function. So if they keep around those cholesterol enzymes longer, then you're gonna be able to make a lot more cholesterol. And that's gonna affect how much cholesterol somebody has floating around in their blood. Yeah, pretty soon. I mean, that's just kind of like, again, the big picture, and that's mostly what I want you guys to take away. I give you guys details because I want to help you guys understand, but for purposes of tutoring, that's kind of like the gist of it. Anyone want to go into any of that in more detail, or do you want to hit something over here? Yes.
So is the splicing of 5-cap for just your eukaryotic, or is it also prokaryotic?
I would say the only thing you gotta remember specific for prokaryotic are these operons.
Okay.
So the splicing is really for eukaryotic.
So number 3 is eukaryotic as well?
Number 3. Yeah, definitely. Yeah, all the rest of these are eukaryotic. When it comes to prokaryotic, that's when I'm talking about operons. Eukaryotic cells do not have operons. That is only for prokaryotic cells. And prokaryotic cells are really simple. This is really their only way to regulate. Again, gray area, but if I'm making it black and white, that's really the only way. The main 95% of the regulation is via operons. Alright, what's next? Purine and pyrimidines. That's good because it's been a while probably since we talked about that. I saw some pictures going around, so I know you guys have pictures of this. I want to keep the physiology stuff up there. Alright, so with purines and pyrimidines, again, you want to start with the starting material and then the finished material, which are the nucleotides. So if we start with, I think I started with pyrimidines in the lecture, so I'll start with pyrimidines again. So what are your pyrimidines? Adenine, guanine, cytosine. So remember, their pyrimidines have the A and G, exactly. Okay, I'm gonna probably do this. But yeah, so you have adenine and guanine. Remember these 15 minutes to like 15 of them. 8 and 2, 22. Finding my video.
Oh, no, you have to put your pyrimidine.
Yes. So when we're starting with pyrimidines, so the key is that you want to remember the starting material, what's going to be adding those nitrogens, which are amino acids, and this one slide is actually got pretty much all information you need, pyrimidines, purines. But essentially, you got to remember the pyrimidines versus the purines. The purines are going to be a little, we can actually put them side by side. So we already said what are the purines versus the pyrimidines. So we need to have the starting material. So what are the purines again? Yes. T and U, and so pyrimidines are your A and T. And when you have your, wait, it switched. No, it switched, it is switched. Now we're doing good. Alright, starting with pyrimidines. So we have 3 amino acids, or 2 amino acids for your pyrimidines. You have glutamine and glycine. And these are what provide the nitrogen, because these are making your nitrogenous bases. So that's what these purines and pyrimidines and purines. We mostly are working about making our nitrogenous bases nitrogen. We need nitrogen. And also remember which ones have a more complex structure. So one of these is, the purines are going to be a 2-ring structure. And the pyrimidines are just a 1-ring structure. And that's going to become important when we have the starting material. So the starting material are these 2 amino acids. We need glutamine, we need aspartate, and we're also going to need carbamoyl phosphate and carbon dioxide and an ATP. But I'm not going to bother with that much. But that's the starting material. Versus purines are going to need those 2 amino acids. So 1, 2, they're going to need glycine, and they're also going to need ribose 5-phosphate. That's their starting material. The ribose 5-phosphate is already a ring. So that's why we have 2 rings for purines. So purines are 2-ring structures because we start out with a ring already. And then they build a second ring. I totally drew that ring wrong, but that's okay. So that ribose 5-phosphate is specific for purines. They start out with the ribose 5-phosphate, and along with these amino acids, glutamine, aspartate, and glycine, that's going to be built on that ribose into a second ring. And both of these are going to require folate. So folate is necessary to make your nucleotides, purines and pyrimidines. And this is the starting material. And what we end up making are your nucleotides. And your nucleotides have the full structure. So nucleotide is the nitrogenous base, and the phosphate groups, and there's a sugar in here too. So CTP, UTP, TTP, these are, and sometimes you'll see them instead like CMP, UMP, TMP. Either way, they're still the nucleotides. It's just one has 3 phosphates and the other one has only 1 phosphate. And I don't care if you remember which is which, it's just these are your nucleotides. Nucleotides, if you recall, I should have started with that. Nucleotides have a nitrogenous group. They have a phosphate group, and they have a ribose, which is a sugar. That's what nucleotides contain. And so ultimately, you start with this material, you're going to be adding some phosphates, so you're going to need some ATP, you're going to be adding extra carbons, you're going to need some extra carbon dioxide, but ultimately, you start with this stuff and you can make these guys. So you will be adding some carbon dioxide and ATP for both of them. And these guys are the same thing, so you start with this, plus you're going to have some carbon dioxide, plus some ATP, and you're going to end up with ATP and, or AMP and GTP, GMP, which are your nucleotides. That's the ending material. And now that you have your nucleotides, you can use these 2 nucleotides to make DNA and RNA. Which is where we pick up the next lecture. Oh wait, we do have to go through the breakdown too, sorry, I forgot about that. So the breakdown of pyrimidines when we break them back down, it's kind of what we already talked about with the breakdown of amino acids. The nitrogen is going to be incorporated into urea. And so nitrogen is going to be ammonia, NH3 is ammonia, and it's going to be incorporated into urea. And what happens to urea? You excrete it, it basically you put it in the blood, it goes to the kidneys, it gets excreted in the urine. That's really the breakdown. And the carbons can be recycled, exhaled, lots of things you can do with the carbons. So it's a pretty simple breakdown for the pyrimidines. If you were to break down the purines, so when you break down your DNA and your RNA back down into the purines are going to be converted into xanthine, which is going to be converted into uric acid. And the uric acid is going to be excreted in the urine too. So purines have a separate path. They have uric acid. And what happens if uric acid builds up? Gout. So if this builds up, it is not excreted properly, you end up with gout. And it's typically a result of not overproduction of uric acid, but our bodies, some people are predisposed to not excreting it very well. And when you can't excrete it very well, when you can't get it out to the urine, then it's going to build up and form these crystals in your joints, and that causes inflammation, gout, and a lot of pain. But that's the breakdown. So purines have a slightly different pathway. But DNA, RNA, the pyrimidines of DNA, RNA just essentially ends up being converted into urea. The nitrogens are just made into urea, excreted, just kind of like amino acids. So obviously a lot of details in the middle there that we don't focus on. That's the beginning material and then how we break it down.
What is that beside purines? Purines and something, and then uric acid. Xanthine.
It's just an intermediate. Not even that interested in xanthine, but it's just, I guess I remember it, so I wrote it down. But the main takeaway, purines are converted eventually into uric acid, and that uric acid can be excreted in the urine. Alright. Let's see, we've done gene expression. We've done nucleotide metabolism. Okay, that's good. The other stuff is easy. Transcription, that's good too, because we can go over that one real quick. Did everyone get what they needed from this? Yep.
Wait, oh, a second, sorry. Oh, you have it? Okay, hold on a second, I'm gonna have it anyway. Thank you, I appreciate it. Sorry.
Alright, so, when it comes to transcription translation, so this one, again, I think I want to say it's so simple. So like DNA replication, there was so much going on. It seemed like we should have a lot more going on with transcription and translation. But we don't. It's, we're going to start with RNA transcription. So
Yeah,
we'll
I
start
forgot
with
the peppermint
DNA.
tea, sorry about that.
And DNA is going to be transcribed into messenger RNA. So this is transcription. And this is, we make the mRNA from DNA. And transcription is going to take place in the nucleus. Because that's where it takes place, is in the nucleus. That's where your DNA is. And there's one primary enzyme that's going to be doing this work. It's going to be RNA polymerase 2. initiation, elongation, and termination. It's those steps, but ultimately initiation. So when we have DNA, so this is going to be the DNA, and this is going to be the gene of interest. Then we have upstream of that this area called the promoter region. And that promoter region has the ta-ta box. It's just a long strand of T-A, T-A-A, just a big long strand of TAs. It's called the ta-ta box. And that is actually necessary for RNA polymerase II to bind. So essentially at this ta-ta box, you're going to have these transcription factors bind. So TF2. There's lots of these transcription factors, and they have to bind here at that ta-ta box. And these guys have to bind first in this promoter region in order to recruit RNA polymerase II. And that's why they're called TF2, because they're recruiting RNA polymerase II. And they have to bind first. Otherwise, you are not going to have transcription. So transcription factors were part of that transcription regulation. So pre-transcription regulation, transcription factors have to bind first. Otherwise, you cannot get transcription. Without TF2s, RNA polymerase will not be recruited. You're not going to make an mRNA transcript. Because this is the whole goal. You want to make a transcript of that gene. So once RNA polymerase binds, so when RNA polymerase binds, it can actually make the mRNA transcript. It's going to be a copy of that DNA. And it's based on the codes. So remember, DNA is going to be in triplets. So you have C, C, A, G, T, C, A, T. 1, 2, 3, 4, 5, 6. We'll add another G here. And so this is, if this is the DNA, then what RNA polymerase is doing, it's got this one now. So what is going to be C? If the DNA is C, what is the mRNA going to be? G, G, U, C, A, G, U, A, C. And that's what RNA polymerase is doing. It's building the mRNA from copying the DNA. So that this whole thing is going to become this mRNA transcript. And these are in codons. So these are the codons that match the DNA triplets. And that's what mRNA polymerase II is doing. That's going to be the tRNA in translation. We're not there yet.
Can we get like 50 of those questions?
Absolutely.
Just
Hopefully you get, do you guys have this in a biology class? If some of you have seen this more recently, maybe some of you. But you probably saw that, like, you didn't see fatty acid synthesis was probably totally new, whereas this, hopefully you've seen a little bit of this at some point. But that's what RNA polymerase is doing, and that's initiation. You have the transcription factors bind, the ta-ta box, RNA polymerase is recruited. You can make the mRNA transcript. That's transcription. That's elongation, and eventually we get to this, then we're to stop, and that's where we have this row, I hate the name of this one, row independent termination. That's where you have in the DNA this long strand of C, C, G, 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 Cs, and they basically create this hairpin loop, and that causes the RNA polymerase to fall off, and that's termination. So it's called row independent termination, which is, I don't know. It's called this name because there is a row dependent termination in prokaryotic cells, but that's one of the main ways that there's lots of ways it can be terminated. This is just an example of ways that eukaryotic cells will terminate RNA transcription. And that's the only way that I have in my slides, so that's just an example. The GCs will come together and they'll create this loop because they are attracted to each other. So G's and C's are attracted to each other. If the mRNA makes, so this is the mRNA, it makes that loop at the end, causing the RNA polymerase to fall off. And that's termination. That's transcription. Oh, then we have the pre-mRNA, actually. And what happens to the pre-mRNA? So we have this strand of mRNA that's got all of these exons and introns. Which ones are spliced out? Did you guys say exons? Intuitively, I want to say exons, but the EX stands for express. Introns are the ones that are cut out.
So they're trying.
They're trying to make this hard. I know, don't second guess. Exons means express. I know, it seems like exit, like you want to get rid of them, but that's
Yeah, I
not
thought you had said that it can only do it on the outside of the DNA, not on the
Oh,
inside.
DNA exonuclease activity. That's the DNA proofreading with DNA replication, which we haven't really discussed yet.
We're getting there.
Yeah, you guys will get there, and next week it's all review, so we can talk about this again in class. You guys are really smart for coming. It is gonna serve you, because next week when we review, this will even be more familiar. Yeah, we haven't done translation yet, but if we are good with transcription, we can either do DNA or we can do translation. Let me get out pictures.
I have one question, where you were doing it,
The hairpin loop.
hairpin loops, something gets cut off, you said that really fast.
So basically,
End of that.
at the end of the gene, there's oftentimes these long chains of C, 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 G, 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 whatever. And these basically, those C's and G's become attracted to each other and create a hairpin loop. So the mRNA itself will make a hairpin loop. And when that happens, that causes RNA polymerase to fall off. That terminates RNA transcription. Yeah, that's the termination.
What makes the hairpin loop from the attraction to the
Yes.
Is there like a word like a stop codon or stop?
That's translation. I know, you're just learning this the first time, it's cool, but let me just briefly do translation before we get into DNA, if we have time for DNA. It's 12:50. You know what, my fifth students are gonna hear. It's gonna be fun. Alright, so just briefly with translation, first off, where does translation happen? In the nucleus or in the cytosol? Cytosol. So the mRNA is gonna have to swim out of the nuclear pore, go into the cytosol, and essentially you're gonna have ribosomes, and which are the really important component. You cannot translate without ribosome. You're gonna have the mRNA, and you're going to have tRNA, which is carrying what? tRNA is carrying what? The amino acid. So the tRNA is carrying the amino acid. So basically, you have to match the codon of the mRNA codon. And what's the start codon?
AG.
AUG, which codes for what? Methionine. And then what will have to be the tRNA anticodon that has to match that AUG? It's no A matches U, A, C is the anticodon that this tRNA is going to match, and then your first amino acid will be methionine that it's carrying to this growing new polypeptide chain. So the whole reason for translation is we're trying to make a polypeptide chain from the mRNA. So we're making a polypeptide chain from the mRNA. basically, we're gonna have a stop codon here, and what's the stop codon I told you guys have to remember? UGA, because that's where I went to undergrad. That's the only one I remember. And that is where the stop codon comes in. So the stop codon is necessary for translation. That's how we terminate translation, is with the stop codon. And there's two others, but I never remember what they are, so I told you guys you don't have to remember them.
It's UAG,
You remember them?
and the other one is UAA, I think.
I'm gonna double check his work. But I don't know them, so I
I
can't.
know UAG is certainly one.
Yeah, you are away anymore now.
Yeah.
Oh, there you
UAA, yeah.
go. Oh,
UAA.
there's another way to remember this.
And your protein subunits are all wrongly drawn. The 30A and they're just wrong size, and we have very much to correct you. I'm just messing around. I apologize. I'm so
It's
sorry.
true, like they do come in sizes, guys. He's making it harder for you though.
If it were me, I'd make him memorize the entire codon. I would make him memorize the entire
Which one?
genetic code. Yes.
That was bad.
See how easy I am.
So, okay, so when it's coming, the mRNA, as it's coming through ribosomes, right?
Oh yeah.
Okay, so the AUG is what?
AUG is the codon. So the mRNA has the codon. The tRNA is the anticodon that has to match the codon. Yeah, the tRNA is the anticodon. So this was the anticodon. Whereas the AUG is the codon of the mRNA. Yep.
So UAC is the.
Yeah, and I don't care if you remember the UAC, but you should be able to figure it out. If AUG is the codon, then the anticodon is UAC. That's what he was saying. He would make you guys memorize them. But he was joking. But no, you don't have to memorize those. The only one you have to know is the start codon, which codes for time. Just
Chaperones, what does that relate to? Is that transcription or
don't worry about chaperones. So chaperones help with protein folding. So I can't remember, that might be part of my slides and I didn't talk about it, but yeah, so basically, yeah. I mean, I would say for the boards, yeah, I talked about it in Biochem 1. And so when it came to folding, I told you guys don't worry about the folding part because we covered that in Biochem 1. So you still need to know for the boards, protein chaperones, like heat shock proteins or chaperones that help fold. So, but for my exam, no, not this coming exam. Proteasomes are in biochemistry too, so you need to know those. Proteasomes, lysosomes. Yeah, yes.
Real quick, I just want to make sure, so we don't need, DNA forms like alpha, beta, gamma, eta.
Right, all of those are eukaryotic DNA polymerases. They're much more complex. We're sticking to the prokaryotic polymerases. DNA polymerase 3 makes the new DNA transcript. DNA polymerase 1, that's the one that's going to get rid of the RNA primers and replace them with DNA. And that's what we didn't talk about yet, but we can talk about that next week too. When I go over DNA. Yeah, that's good.
Are you gonna do, so I know we're reviewing class on this, are you going to record a session?
I'll still be here next week. Yeah, I'll still be here at noon. If you guys want to show up, then we can do it again. I'll get out of these pictures. And I might be too late.
Sorry, you look great. I need to drop by my apartment real quickly to grab the flashcards, and then I'll be at the library.
Does the DNA replications, because the actual work of making the lead
Well,